Hey guys, welcome back to my channel. In this video I'm going to be going over amino acids. Amino acids in general you would link to proteins because we hear about the important amino acids in protein foods. And we are correct, amino acids are used to make proteins. But in this video I'm not going to be going over that. That process involves translation, and that's going to be a separate video. In this video I'm going to be going over the structure of an amino acid. So let's dissect this word first. So amine refers to an amine group. So it's got something similar to ammonia. It sounds a lot like ammonia. An acid, this part refers to carboxylic acid. So this is the way to remember. So we think of ammonia and then carboxylic acid. When you think about ammonia, you think of nitrogen and hydrogen, right? Well, let's think of that instead of ammonia. Let's think of nitrogen and hydrogen. And the structure of an amino acid looks like this. So on this end we have this what we call an amine group. The chemistry students here who've done topic 10 should be able to recognize this. Similarly, we have something here called the carboxylic acid group, which I'm actually forgetting the right name for it, but it might be that. But there's a C carbon double bonded to an oxygen and double bonded to an alcohol group to form a carboxylic acid. This middle carbon here is going to be bonded to a hydrogen and an R group. R group is the variable group, and this determines what the amino acid is going to be. So identity of the amino acid. So it could be methionine, isoleucine, etc. So if we have two amino acids side by side, what they can do a nifty trick where these two connect. Let me just do a plus. And what we get is diamine. Sorry, give me a sec. This is gonna give us so we've done this so far. Now we had this OH and this H combined, and when two substances combine in this way to form one substance, we call that an anabolic reaction. Anabolic, you can think of anabolic steroids, they help build up muscles. And this H, another H and an O, is quite similar to H2O. So this forms water, and similar with rain, where we call it condensation, we call this a condensation reaction. So we have an amino acid and an amino acid, we form a dipeptide. And the dipeptide is formed with what we call a peptide bond. This is what the new structure is going to look like. And what we have here is something we call the peptide bond. So the peptide bond is going to be linked with the rightmost carbon and the leftmost nitrogen. So that's how you remember where the peptide bonds are going to be. And since we have this dipeptide, we can form a longer tripeptide and a quadpeptide, or whatever it's called, tetrapeptide, I have no idea. But this chain can go on, and that's how we get the primary structure of a protein, right? So the primary structure of a protein is the sequence of amino acids, and this formation of peptide bonds to form dipeptides and longer chains of amino acids is the first step in forming our protein. Let's do a couple practice questions. In this question, we are asked to identify where the peptide bonds are in this polypeptide. A polypeptide simply means many, peptide means amino acid, so many amino acids linked together. Here I can count 1, 2, 3, 4, 5 amino acids linked together. And as you might be seeing, there's no R group mentioned here. Instead, they've actually substituted something for the R group. There's a hydrogen here, CH3, CH3, CH2, H, and another hydrogen. So those are the R groups that are going to be there. So just because there's no R group in a diagram does not mean it's not an amino acid. So previously we said that an amino acid has the peptide bond at the link between the right carbon and the left nitrogen of a new one. So if we identify this is a peptide bond. This is a peptide bond. This is a peptide bond. This is the peptide bond. There are 4 options for peptide bonds. We only need to label one, so let's just pick this guy. And that's our question for the 1 mark. Next question, outline the primary structure of proteins. The primary structure I mentioned previously is the sequence of amino acids in a polypeptide. So primary structure is the sequence of amino acids in a polypeptide chain, and this actually determines the properties of the protein to a large extent, because the secondary, tertiary, and quaternary features of the protein are going to be determined by the identity of the amino acids. And also, if methionine is followed by isoleucine and followed by leucine, that's not the same as leucine being followed by isoleucine and methionine. So the sequence of these amino acids being changed makes the properties of the final protein become different. So other than that, guys, this is going to be the end of the video. Hope you guys learned something new. And as always, feel free to comment down below any video suggestions you want me to do. Any topics you guys want me to cover, I'll be happy to. And like and subscribe, please. And yeah, enjoy.